The drop of sap descends in geological slow motion, a gilded tear sliding down the bark of a Cretaceous conifer. It envelops a female mosquito, its final meal of reptilian blood still warm in its abdomen, its last frantic buzz silenced forever. For the next 66 million years, this becomes a silent tomb of hardened resin, a perfect golden stasis. Inside, the mosquito is an immaculate relic. The blood cells of a Tyrannosaurus rex, microscopic and suspended, begin their impossibly long wait. Within each cell, the blueprint of a lost world is coiled into chromosomes, a spiraling library of adenine, guanine, cytosine, and thymine. This is the code for scaly skin, for hollow bones, for a 2-ton skull designed to crush its prey. It is the ghost of a giant. For millennia the information remains, but it is not immortal. Deep time is a relentless thief. The faint hum of background radiation from the Earth itself bombards the helix, shattering its rungs. Water molecules, the universal solvent, creep through microscopic fissures in the amber and perform their slow, inexorable work of hydrolysis, snipping the bonds of the sugar-phosphate backbone. Oxygen, the very element that fueled the dinosaurs' powerful muscles, now attacks the delicate bases, corrupting the code, one letter at a time. The library is not just being closed, it is being burned. What was once a complete set of encyclopedias detailing the construction of a Mesozoic predator becomes a scattering of charred pages, then fragments of sentences, then isolated, meaningless words. By the time a primate with a keen intellect and a polishing wheel unearths this gilded coffin, the blood cells are gone, their membranes long since dissolved, and the DNA, the sacred text of a bygone ecosystem, it is less than dust. It is a chemical echo, a faint whisper of what once was. The miracle isn't that we found it. The tragedy is that it arrived 66 million years too late. To understand the sheer fantasy of resurrecting a dinosaur, we must first appreciate the staggering reality of what a living creature is. An organism is not merely a collection of parts, but the result of an ongoing 4-billion-year-old conversation written in the language of deoxyribonucleic acid, DNA. This is the famous double helix, a molecular ladder whose rungs are made of 4 nucleotide bases. The specific sequence of these bases forms genes, which are the recipes for creating proteins. These proteins, in turn, build everything from the keratin in a T-Rex's scales to the hemoglobin in its blood. The entire collection of these recipes for a single organism is its genome, a biological instruction manual of breathtaking complexity. The human genome, for instance, contains over 3 billion of these base pairs. A dinosaur's would be of a similar magnitude. The process of cloning, as we practice it today, is a marvel of precision, but it hinges on one non-negotiable requirement: an intact, living cell.
The landmark case of Dolly the sheep in 1996 relied on a technique called somatic cell nuclear transfer, or SCNT.
Scientists took a somatic cell, any cell, from the body that isn't a sperm or egg cell, from the udder of one sheep. They carefully extracted its nucleus, which contained the complete, undamaged genome. Then, they took an egg cell from a different sheep and removed its nucleus, effectively erasing its genetic instructions. The nucleus from the first sheep was then implanted into this enucleated egg. With a gentle electrical pulse, the egg was tricked into behaving as if it had been fertilized, and it began to divide, using the implanted DNA as its new blueprint. This developing embryo was then placed into the uterus of a surrogate mother sheep, and months later, Dolly was born, a perfect genetic copy of the sheep from which the udder cell was taken. This process is the gold standard for cloning. Notice the crucial elements. A living, healthy donor cell providing a pristine, complete genome, a viable living egg cell to act as the vessel, and a compatible surrogate mother to carry the pregnancy. Jurassic Park's premise cleverly attempts to substitute each of these components. Instead of a fresh somatic cell, it proposes a blood cell preserved in amber. Instead of a state-of-the-art laboratory, it relies on a mosquito's gut as a perfect biological refrigerator. And instead of a single complete genome, it presents us with fragmented DNA that can somehow be patched and repaired. It is an elegant and seductive solution, a piece of storytelling genius that captured the world's imagination. But when placed under the unforgiving lens of chemistry, biology, and time, this elegant solution unravels at every conceivable point. The fantasy collapses not on a single hurdle, but on a cascade of absolute, fundamental impossibilities. The story is beautiful, but the science tells a different tale, one of decay, complexity, and the irreversible arrow of time. The first and most insurmountable wall that stands between us and a living dinosaur is the simple, brutal chemistry of decay. DNA, for all its power as the molecule of life, is just that, a molecule. And like all complex organic molecules, it is fundamentally unstable. Its existence is a constant battle against entropy. And once an organism dies, it is a battle the DNA will always lose. The primary mechanism of its destruction is its interaction with water, a process known as hydrolysis. This chemical reaction systematically severs the bonds holding the long polymer chains together, breaking the genome into smaller and smaller pieces. Another process, oxidation, damages the individual nucleotide bases, effectively corrupting the genetic letters themselves. Think of the genome as an immense, intricate novel. Hydrolysis is like an acid that dissolves the binding and breaks the book into loose pages, while oxidation is like ink bleeding through the paper, making the words themselves illegible. For years, scientists hoped that certain conditions, extreme cold, dryness, or, as in the film, the protective shroud of amber could halt this decay indefinitely. This hope was systematically dismantled by a groundbreaking 2012 study led by Morton Allentoft and Michael Bunce. They analyzed DNA from the leg bones of the 158 giant moa, extinct birds from New Zealand, with remains ranging from 600 to 8,000 years old. By comparing the amount of DNA degradation across these samples of different ages preserved in nearly identical conditions, they were able to calculate a definitive rate of decay. They found that DNA has a half-life of 521 years. This means that after 521 years, half of the chemical bonds in a DNA sample will have been broken. After another 521 years, half of the remaining bonds will break, and so on. Let's apply this unforgiving number to our dinosaur. The last non-avian dinosaurs died out at the end of the Cretaceous period, 66 million years ago. A half-life of 521 years means that the amount of intact DNA is halved roughly 126,680 times. The resulting number is so infinitesimally small as to be meaningless. It is not a fragment, it is oblivion. The study concluded that even in the most perfect, deep frozen conditions imaginable, a constant -5 degrees Celsius, every single readable bond in a DNA strand would be completely destroyed within a maximum of 6.8 million years. The youngest dinosaur fossils are nearly 10 times older than that absolute upper limit. Amber, unfortunately, is not a better preservative. Early claims of extracting dinosaur-era DNA from insects in amber in the 1990s generated immense excitement, but were later proven to be the result of modern lab contamination. More rigorous studies have shown that amber is not a hermetically sealed environment. It's permeable to water over geologic timescales, and so the slow destructive process of hydrolysis proceeds unabated. What scientists have successfully recovered are magnificent cellular structures, ghost cells where the DNA once resided, but the DNA itself is gone. There is no code left to read. We are staring at an erased hard drive. But let us engage in a colossal act of scientific fantasy. Let us imagine we overcome the laws of chemistry and find a miraculously preserved sample containing not just fragments, but a significant portion of a velociraptor genome, say, 90% complete. This leads us to Jurassic Park's most iconic and misleading sleight of hand. We fill in the gaps with frog DNA. On the surface, it sounds plausible, like patching a tear in a piece of fabric. In reality, this is like trying to fix a corrupted line of C++ computer code by pasting in sentences from Shakespeare. The result would not be a functional program, it would be catastrophic gibberish. A genome is not a simple linear string of instructions read from A to Z. It is a four-dimensional system of staggering complexity. Genes interact with each other in intricate networks. Regulatory sequences, vast stretches of so-called junk DNA, act as sophisticated switches, telling genes when and where to turn on or off during an organism's development. An eye gene turning on in a liver cell would be a disaster. The timing is everything. To build an embryo, thousands of genes must be activated and silenced in a precise, symphony-like cascade that unfolds over days, weeks, and months. Introducing DNA from a modern amphibian, which last shared a common ancestor with dinosaurs over 340 million years ago, is not a patch. It is an active genetic sabotage. The frog's genetic code, its regulatory sequences, and the proteins it produces would be completely alien to the dinosaur's cellular machinery. The dinosaur transcription factors, the proteins that read the DNA and initiate gene expression, would not recognize the frog's promoter sequences, or worse, they might bind to them incorrectly, activating the wrong genes at the wrong time. Imagine the cellular chaos. Genes for reptilian scales are suddenly confronted with instructions for a permeable amphibian skin. The delicate development of a three-chambered reptilian heart is disrupted by signals intended for a different circulatory architecture. The process would not create a fascinating hybrid. It would fail at the first hurdle, the initial cell division. The embryo would collapse into a non-viable collection of cells almost immediately. The gaps in the dinosaur genome are not just missing letters. They are missing chapters, missing context, missing the entire grammar of the book. And trying to fill them with a completely different language doesn't complete the story. It makes the story impossible to read. Let's escalate our fantasy to its most extreme. By an act of scientific divinity, we are handed a perfect, complete, 100% intact Tyrannosaurus rex genome sequence, stored on a computer. All 3 billion base pairs are present and accounted for. The problem of decay is gone. The problem of filling gaps is gone. Now we simply have to turn this digital code into a living, breathing animal. We now face perhaps the most subtle, yet most profound, biological barrier of them all, the translation of information into flesh. A genome is a blueprint, but a blueprint needs a construction crew, materials, and a factory. For an animal, that factory is the egg. Our first task would be to synthesize this genome, building an artificial chromosome. This is something we can theoretically do, though assembling a structure of this size with perfect accuracy is beyond our current capabilities. But assuming we manage it, we would then need to perform somatic cell nuclear transfer. We need to place our synthetic T-Rex nucleus into a compatible enucleated egg cell. But where do we get such an egg? The closest living relatives to dinosaurs are birds. So we might choose the egg of an ostrich, one of the largest available. Here we collide with the problem of the cytoplasm. The cytoplasm is the jelly-like substance filling the egg cell, and it is not just passive filler. It is packed with thousands of vital maternal proteins and RNA molecules. These cytoplasmic factors are essential for orchestrating the very first stages of embryonic development. They guide the initial cell divisions, help organize the body plan, and protect the new nucleus until it can take control of the cell. Critically, these factors are species-specific. They are designed to interact perfectly with the proteins and DNA of their own species. An ostrich cytoplasm is calibrated for an ostrich nucleus. When it encounters the wholly alien DNA and proteins of a Tyrannosaurus, the two systems would be utterly incompatible. The signaling pathways would not connect. The ostrich proteins would be unable to properly regulate the dinosaur genes. The mitochondria, the cell's power plants, which have their own small set of DNA and are inherited exclusively from the egg, would be foreign to the new nucleus. The rejection would be immediate and total. The embryo would arrest development before it even truly began. And even if it could survive this initial cellular mismatch, there is another, phantom layer of information that was lost the moment the dinosaur died: the epigenome. Epigenetics refers to the chemical markers, such as methyl groups, that are attached to the DNA molecule. These markers don't change the sequence itself, but they act like sticky notes, telling the cellular machinery which genes to read and which to ignore. This epigenetic programming is crucial for development, determining which cells become skin, bone, or nerve. It is the operating system that runs the genomic hardware. But this intricate pattern of markers is erased and rewritten with each generation, and it vanishes upon death. Our synthetic T-Rex genome is a perfect copy of the hardware, but it is missing the operating system entirely. We would have no way of knowing which of the 20,000 plus genes should be switched on or off, in which cells and at which times to correctly build a Tyrannosaurus. Without these phantom instructions, our perfect genome is inert. It is a master plan with no one to read it. From chemistry to genetics to developmental biology, the hurdles are not just high, they are absolute. A true living dinosaur is locked away from us forever by the fundamental laws of biology. The failure to resurrect a dinosaur is not, however, an endpoint for human ingenuity. Instead, it forces a crucial clarification. The dream of de-extinction is not dead, but it is being reborn in a more realistic and perhaps more complicated form. We cannot clone an extinct species, but what if we could edit a living one? This is the central premise behind the real-world science of de-extinction, a field focused not on resurrection, but on approximation through advanced genetic engineering. The flagship project in this endeavor is the revival of the woolly mammoth. A team of scientists at the company Colossal Biosciences is not trying to find intact mammoth DNA. They know that's impossible. Instead, they have sequenced the mammoth genome from degraded fragments found in the Siberian permafrost. They are now comparing this ancient blueprint to the genome of the mammoth's closest living relative, the Asian elephant. Using gene editing tools like CRISPR, they are meticulously editing the elephant's DNA in lab-grown cells, Their goal isn't to create a perfect mammoth clone, but a cold-resistant elephant proxy. They are identifying the specific genes that gave the mammoth its signature traits and splicing them into the elephant genome. This includes genes for long, shaggy hair, for a thick layer of subcutaneous fat, for smaller ears to reduce heat loss, and for a type of hemoglobin that could efficiently release oxygen into its tissues at low temperatures. If they succeed, they will have an embryo with a heavily modified elephant genome, one that produces mammoth characteristics. This embryo would then, theoretically, be carried to term in an artificial womb or by a surrogate elephant mother, though the latter poses immense ethical challenges. The resulting animal would not be a woolly mammoth. It would be a new kind of elephant, an elephant-mammoth hybrid, designed to fulfill a similar ecological role. This distinction is profoundly important. It shifts the conversation from a nostalgic fantasy to a serious ecological proposition. The goal of the Mammoth Project isn't to populate a theme park, but to reintroduce a large grazing herbivore to the Siberian tundra. Proponents argue this mammoth steppe ecosystem could be restored, with the animals trampling and grazing habits preventing the thaw of carbon-rich permafrost. Similar projects are underway for other species. The passenger pigeon, once numbering in the billions, is being targeted by editing the genome of its relative, the band-tailed pigeon. The thylacine, or Tasmanian tiger, might be approximated by modifying the genome of a dunnart, a mouse-sized marsupial. But this new frontier raises dizzying questions. Is it right to create a proxy species and release it into a world that has changed since its original counterpart vanished? An animal is more than its genes. It's a product of learned behaviors, gut microbes, and its environment. How would a lab-grown mammoth elephant learn to be wild? What unforeseen impacts would it have on modern ecosystems? And there is a deeper philosophical question. In our rush to undo our past mistakes, are we playing God in a new, more precise way? We are not reanimating the dead, but engineering new forms of life to suit our own purposes, however noble they may seem. Jurassic Park's fantasy was a warning about hubris in the face of nature's power. The reality of de-extinction is a more subtle story, one about our responsibility as we gain the power not to resurrect the past, but to rewrite the future of life itself. The dinosaurs remain impossible, but the moral and ecological dragons of genetic engineering are very, very real. The final, insurmountable barrier to recreating a dinosaur is not in the end one of technology or chemistry. It is one of identity. Time does not just degrade information, it transforms it. Life is not a static blueprint that can be paused and restarted. It is a relentless, unbroken river of ancestry, a 4 billion-year chain of inheritance linking parent to child, species to species. When a species goes extinct, that chain is broken forever. A link is not just lost, the river itself is diverted. Recreating a Tyrannosaurus rex by assembling its ancient DNA is like trying to recreate a forgotten language by analyzing a single shredded page of its literature. You may be able to piece together a few words, but you will never recapture the grammar, the syntax, the culture, the songs, the soul of what it was. You will have a lifeless caricature. And here lies the ultimate revelation, the paradigm shift that reframes this entire discussion. The obsession with bringing dinosaurs back is born from the belief that they are truly gone, that their world is a sealed and inaccessible past. But this is the greatest misconception of all. We don't need to look for dinosaur DNA in the hardened sap of ancient trees, because that DNA is not lost. It is all around us. It has been edited, repurposed, and refined by 66 million years of evolution, but its lineage is unbroken. Dinosaurs never truly vanished. They learned to fly. Look at a cassowary as it stalks through a rainforest. See the scaly, three-toed feet, almost identical to those of the velociraptor imprints found in stone. Look at the S-shaped curve of its neck, a classic theropod posture. See the predatory intelligence in its gaze, the direct, unbroken inheritance of a line of bipedal hunters. Listen to the deep, resonant boom it makes, a sound that echoes the calls that once filled Mesozoic forests. This is not a replica. This is not a proxy. A bird is a living, breathing dinosaur. An eagle, stooping from the sky with its talons outstretched, is carrying the genetic legacy of the most successful predatory dynasty the world has ever known. The wonder of Jurassic Park was seeing a T-Rex walk the earth again, but the truer wonder is recognizing that its direct descendants, numbering in the billions, share our world today. Their DNA is not a static relic, it is a dynamic, evolving masterpiece. The quest to resurrect a lost world distracts us from the miracle of the one that survived. We don't need to rebuild the past, we only need to open our eyes and see it thriving, in feathered